Good afternoon. I hope everybody enjoyed their break and was able to check out the Network Lounge during that break. Our first presenter of the afternoon is Nate Ellert of the Canola Council of Canada. Nate will be talking about canola research programs. And if you have questions for Nate, please type them into the Q&A box on your screen. We will then address these questions at the live question and answer session.
Hi everyone, my name is Nate Ellert. I'm an agronomy specialist with the Canola Council of Canada. And today I'm just going to be briefly speaking on our CARP and CAP research programs. So what is CARP? CARP is a canola agronomic research program and is funded by the 3 provincial canola grower organizations, the Manitoba Canola Growers Association, the Saskatchewan Canola Development Commission, and the Alberta Canola Producers Commission. And what the Canola Council of Canada does is we administer this research program, and we bring these 3 grower group organizations together, and we mediate the process of who's funding what. And what happens is sometimes 2 of the grower group organizations will fund a project, or sometimes all 3 of them will, or sometimes just 1 of them does. Further to this, the agronomy specialists all provide feedback for projects that coincide with their area of expertise. And this just gives, this eases the decision-making process a little bit. It gives the research committees within each grower group organization just some more information and a perspective, from the experts on our team. CARP is a 1-year funding cycle program. So every year we are receiving new research proposals. And the following year, some of this research begins. So it's a very quick turnaround. The key findings from these projects are what we use in our extension messaging. And this can translate into agronomic recommendations, or what is currently being worked on, what's coming down the pipeline. Further to this, a lot of the key findings can be found on our Canola Research Hub. So this is our database where we store all of the research that has been done or is actively going on. And then a lot of the key findings we use as resource material in Canola Council publications such as Canola Digest, Canola Watch, and the Canola Encyclopedia, which is this massive database on our website of the best agronomic practices and what to look out for diseases and fertility recommendations and things like that. So here are some examples of 2 recently completed CARP studies and 2 CARP studies that are ongoing right now. So the 2 completed ones that I have listed here are both from the University of Manitoba. One is looking at slow release monoammonium phosphate and struvite to improve phosphorus use efficiency and to reduce seedling toxicity in canola. And that's being done by Dr. Zamwea in the Department of Soil Science. Also in the Department of Soil Science is Dr. Tenuta and he recently completed a CARP-funded study on canola response and minimizing nitrogen losses in a 2-pass seeding fertilization system with different placements in Manitoba. So those are 2 recently completed studies, and then I just wanted to touch base on 2 studies that are actively going on. So Dr. Tenuta again is looking at optimal source, placement, and timing for yield and the reduction of greenhouse gases in canola production. And Dr. Malicki with Agriculture and Agri-Food Canada is looking at how we can reduce seed row toxicity in phosphorus fertilizer and canola. So what's next for CARP? Like I said, it's a 1-year funded, 1-year funding cycle. So right now we're reviewing the full proposals for 2021. The Canola Council Agronomy team right now is going through these research proposals and providing feedback, which will end up in the grower group organization research committee's desk, and they can use to help with their decision-making process. Moving forward for CARP 2022, these letters of intent are usually called for in early summer and are due in the 1st or 2nd week of September. For more information on CARP, you can go to our website. The link here is on the slide, or you can contact myself. My email is on our website or also at the end of this presentation. So moving forward, what is the Canola Agri-Science Cluster, or the Canadian Agricultural Partnership, which is what CAP stands for. So what this is it's the same funders from before, plus the Canola Council of Canada and Agriculture and Agri-Food Canada, which is, of course, the federal government. And what goes on here is these 5 organizations get together for the Canadian Agricultural Partnership, and it's similar to CAP to CARP, as in the funding comes from these 3 grower groups, which is ultimately funded by farmers, and then this is just a top up. And the other big key difference is the Canadian Agricultural Partnership is a 5-year funding program, so it's quite a larger time frame. But this also means that there's also a lot more money for it. So in the canola cluster, there's just over $25 million, that is split up between research projects over 5 years. And again, what we do with these key findings is this translates and it helps with our extension messaging. It's put up on the Research Hub. We use on Canola Digest, Canola Watch, and Canola Encyclopedia. So again, just wanted to touch base on what has been done, just to provide some examples and some context. So Dr. Grant with Agriculture and Agri-Food Canada out of Brandon looked at the impact of traditional and enhanced efficiency phosphorus fertilizers on canola emergence, yield, maturity, and seed quality. Dr. Moulin, also with Agriculture and Agri-Food Canada, recently completed a study on variable nitrogen fertility management of canola at the field scale based on analysis of yield maps and spatial and statistical variation of soil test nitrogen and phosphorus. 2 ongoing fertility-related CAP studies are by Dr. Ma and Dr. Vail, and I believe they're talking about these immediately following this presentation, so there's not really any reason for me to touch base on them, and we can just wait for the experts. So what's next for CAP? CAP is currently in year 3 of 5 of its funding cycle, so CAP ends at the end of March in 2023. Right now, we're in the planning stages for the next funding cycle, so identifying our research priorities and just getting our ducks in a row for, when the next funding cycle and program is announced, we can hit the ground running and we're ready, to be as efficient as we most possibly can be when this happens. So the takeaways, CARP and CAP are both farmer-funded research programs. If you have a research idea, farmers are the ones that are out in their fields every day. same with agronomists, and your perspective and what you are seeing is likely different than what researchers are seeing. So, I would encourage you to, if you have any ideas like, oh man, this really could be researched upon, to reach out to either your grower group research committee for them to bring it forward. You could contact myself, you could contact an agronomy specialist. Maybe this research has already been done. Maybe not, maybe it needs to be done. So, there's different avenues to explore there if you have a research idea or a question. And I also just wanted to highlight that CARP and CAP are not exclusively funding fertility-related projects. They fund all sorts of different projects relating to canola production, whether that's agronomy or end use, or etc. So here are some resources. I went over these. I just wanted to stick these on a slide so you had the links, but there's a link to the CARP page, the CAP page, and then all of our publications and our encyclopedia. And then if you're curious on more extension messaging, that's when you would contact one of us, one of the agronomy specialists here at the Canola Council. And that's all I have for today. So I'm on for the live session, but I would encourage you to reach out to me with any questions you might have. Thanks.
Our next presenter is Dr. Bao-Luo Ma of Agriculture and Agri-Food Canada. Dr. Ma will be presenting on his ongoing research project. If you have questions for Dr. Ma, please type them into the Q&A box on your screen, and we will address them during the next live question and answer session.
Thank you. Good afternoon. Today, I will talk about improving nitrogen use efficiency and soil sustainability in canola production across Canada. In this presentation, I will give you a brief background information, followed by the objectives, describes the experimental procedure, and show you some key results and the preliminary summaries, followed by a slide to list our future work. As we know, nitrogen is the most limiting nutrient in canola production. Use of nitrogen fertilizer can increase canola biomass, yield, and seed quality. But inappropriate use of nitrogen fertilizer can lead to increased cost for the grower, reduced seed oil concentration. For example, at the optimal nitrogen application, seed oil concentration can be somewhere between 45 to 46%. Too much nitrogen can reduce this by 1 to 2%. Too much nitrogen also increases the risk of lodging, limiting yield potential, and causing negative impact on the environment. Therefore, improving nitrogen use efficiency can not only benefit the short-term profit of canola growers, but also improve long-term soil health and environmental sustainability. Keep this in mind, we conducted 2 field experiments with 4 objectives. First, to assess agronomic and economic responses of canola hybrid cultivars to nitrogen fertilizer application. Second, to improve nitrogen use efficiency, crop productivity, and lodging resistance of canola crop through best nitrogen management practices under different conditions. In addition, we want to identify root architectural traits and investigate the taxonomic and functional response of the soil microbiome to nitrogen management. Due to the limitation of time, I will focus my talk to the first 2 objectives. We conducted 2 field experiments across 8 locations in Canada. That's 2 in Alberta, AFC Beaver Lodge, Oates College, 3 locations in Saskatchewan, that's AFC Medford, Scott, and Swift Current. 2 locations in Manitoba, that's AFC Brandon and Carmen of the University of Manitoba. In Ontario, Ottawa, that's where my research station located. The first experiment was arranged in a randomized complete block design with 4 replications at each site in each year. We tested 2 current hybrid cultivars in response to the timing and the rates of nitrogen fertilizer. At the pre-plant, we have 0, 50, up to 200 kg per hectare. We compare with the split application that's on the basis of 50 kg per hectare plus 50, 100, or 150 kg nitrogen per hectare, top dressed at the 4 to 6 leaf stage. The second experiment was designed to determine nitrogen requirement of canola in different cropping systems. We tested 3 rotations, that's intensive, which is canola, canola wheat, and canola, compared to 2-year rotation, that's wheat, canola, field pea, and canola, or 1 canola in 4 years, wheat, flax, field pea, and canola. During the canola year, we tested 5 levels of nitrogen. We determined the soil, crop growth parameters, yield, and nitrogen uptake. For the soil, we determined soil texture, soil pH, soil organic matter, soil mineral nitrogen concentration at the top dress and at the post-harvest stage. For the crop, we determined phenology, standard counts, plant biomass at the early flowering stage and at maturity. At maturity, we determine the yield components, and the nitrogen concentration in the seed and the straw. With this data, we calculate agronomic nitrogen use efficiency, which is use the difference method, the yield with nitrogen application minus yield without nitrogen application divided by the amount of fertilizer applied. We determined maximum economic rate of nitrogen, mer value, using the quadratic response. We taking the first derivative to get the plateau yield and at which the maximum nitrogen required. One important consideration is the economy. So we consider the nitrogen cost and the yield price value. So we introduce the B factor into the model to get the mer value. We measured a bunch of root parameters. For example, we're using the beacon precision meter to determine electrical capacitance. Use destructive sampling, we determine root length, root surface area, and root volume, and the biomass. With this information, we can better understand how root functions. For the another area, we want to determine the lodging risk. We're using the 3-point bedding test to simulate stem lodging and using the smart screwdriver to simulate root lodging. We dissect the stem base to determine the anatomical traits. In the following few slides, I will show you some key results we found in this study. From the first experiment, we observed several important points. First, responses of canola yield to nitrogen application varied among sites in both years. There are 3 cases in the first year and 7 cases in the second year. We found yield increased with the rate of nitrogen application. In some cases, split nitrogen application had higher yield than the same amount of nitrogen applied only at the pre-plant. In most cases, there was no nitrogen by hybrid interaction, meaning they had similar response to nitrogen. The estimated MER value was on average 170 kg per hectare, meaning the yield at the MER varied from 1.4 to 4.8 tons per hectare. This means for each kilogram nitrogen per hectare applied, canola yield would increase by 10 to 14 kg per hectare. This slide shows two examples. Saskatchewan, there are 3 locations we calculate the average MER value, so that's for pre-plant application that requires slightly higher MER value, but with slightly lower yield. Therefore, the split application had a higher yield with slightly smaller MER value but a larger yield increment. In contrast, at the Alberta 2 locations, where the yield increment was very similar for the 2 application methods, but for split application, the seed yield was higher than pre-plant application and therefore required a slightly larger MER value. We found canola yield was very strongly and positively correlated with the plant height, with the plant biomass at the early flowering stage and at the maturity. Yield was correlated with the number of branches and number of pods per plant at most locations. When leaf chlorophyll measured with SPED and the canopy reflectance expressed as NDVI, as normalized difference vegetation index, we found these values were closely correlated with yield at the selected site. From the second experiment, we found canola following wheat had a higher seed yield than intensive canola production. So the rotational canola also had a higher nitrogen use efficiency in 5 out of 7 locations. And therefore, canola in the rotation will require a smaller MER but with a higher estimated yield compared to intensive canola production. In summary, our data shows canola yield positively responded to nitrogen application rates, with a better response to the split nitrogen application method in some cases compared to the same amount of nitrogen applied only at the pre-plant. Under favorable growing conditions, the estimated MER, that's maximum economic rate of nitrogen, averaged at 170 kg per hectare, with achievable yield up to 4.8 tons per hectare. Canola growing in rotation with wheat had generally higher yields with a lower MER requirement than intensive canola production. Our data clearly indicate a need to develop region-specific nitrogen recommendations to achieve both profitable and environmentally sustainable canola production in Canada. There is a large variation in the environment. Therefore, it takes multi-site years of field tests to develop site-specific recommendations. We plan to using modern tools such as soil spectra and crop indicators that will help us better understand soil nitrogen dynamics and develop sensitive models based on crop needs. We want to incorporate the soil, plant, and weather data into the multivariate analysis that will help us develop models for predicting the site-specific, most optimal rate of nitrogen requirement by canola. I would like to take these opportunities to acknowledge Canola Council of Canada who set up the research priority and led this cluster project. As mentioned, there are many AFC scientists and university professors who participate in this collaboration research. At each location, there are many technical support staff and students who provided excellent research assistance. Thank you.
Our next presenter is Dr. Sally Vail from Agriculture and Agri-Food Canada. Dr. Vail will be presenting on her ongoing research project. If you have questions for Dr. Vail, please type them into the Q&A box on your screen, and we will address the questions during the live question and answer session following Dr. Vail's presentation.
Thank you to Warren and other members of the Canola Council for the invitation to share the vision and objectives of our canola cluster project, where we are studying different aspects of nitrogen uptake and remobilization towards establishing a better understanding of how to improve canola NUE. The long-term goal of this work is developing genetic resources and tools for breeding of cultivars, or genotypes, that better fit into different G by E by M, or genotype by environment by management systems. So this is not only to reduce economic nitrogen losses for growers, but also environmental nitrogen losses from canola rotational cropping systems. I'd like to start out by introducing the team of researchers who are working on this project. From the University of Saskatchewan, Rosalind Buchert is the crop physiologist who is leading the intensive field-based sampling and associated physiological analyses. Also from the U of S is Melissa Arcand from the Soil Science Department, who is looking at soil nitrogen cycling and providing her expertise to the field experiments. From AFC, Raju Solikanahalli is a plant physiologist who is examining nitrogen uptake and assimilation in plant organs. Also from AFC, Isabel Parkin is a Brassica geneticist, and Steve Robinson has extensive experience in stress physiology and bioinformatics. And Steve and Isabel are working together on establishing high-throughput phenotyping systems for brassicas under controlled conditions. What I expect has been highlighted in the presentation so far today is the complexity of the N cycle in cropping systems. So further adding to the complexity around NUE is different ways in which it's measured. For example, agronomic NUE is based on seed yield measurements given fertility treatments, which is different than crop physiology-based methods that can account for total plant nitrogen uptake and subsequent movement within the plant throughout the growing season. Overall nitrogen use efficiency can be subdivided into nitrogen uptake efficiency and nitrogen utilization efficiency, where nitrogen uptake efficiency is the effectiveness of N acquisition from the soil by roots in the rhizosphere, and nitrogen utilization efficiency is utilization of N once it's in the plant. Existing literature and ongoing work in Europe and Asia have shown that natural variation for both uptake and utilization efficiency has been identified and characterized in winter and semi-winter Brassica napus, yet this variation has not yet been comprehensively studied in Canadian cultivars, in germplasm, or in spring types in general. In order to set the stage for the specific experiments in this project, I wanted to first introduce the 3 different collections of germplasm that were used for the different studies. The first leveraged an experiment you heard about from Dr. Ma, which was experiment 1, where the yield response to timing and rates of nitrogen fertilizer was assessed on 2 current commercial hybrids. Secondly, a Canadian historical panel of rapeseed and canola cultivars was established, which contains open-pollinated cultivars starting with Argentine, released almost 80 years ago, to 2002, along with hybrids released 2007 through 2017, plus a few breeding lines that have been developed at Ag Canada over the past few decades. The historical panel is being used for a large field trial, and a subset of plots from this trial are being used to look at root system architecture and root microbial soil interactions. And under control conditions, the historical panel is being used to address specific physiological hypotheses by Raju. And finally, Steve and Isabel are using the diversity panel to develop high-throughput phenotyping systems. The nested association mapping, or NAM population, is a structured population that's designed to study and develop new germplasm through wide crosses with a parental panel of founder lines. And these founder lines represent comprehensive diversity of accessions from all across spring Brassica napus, including lines not only of Canadian origin, but also worldwide origin, such as European, Australian, and Asian descent. Building on the Agronomy CAP project trial, which was designed to look at rate and placement response, this experiment's objective was to quantify how much N commercial hybrids are taking up from the soil across the whole plant, including not only the seed, but all the plant organs and across various growth stages. For this, Dennis Sulik, a master's student, intensively sampled at Scott and Melfort locations in 2019 and Saskatoon in 2020. This slide captures the plant sampling and processing that's involved in this type of work and the intensive and time-sensitive partitioning of the plant into leaves, stems, pods, and seed, and the additional measurements and processing prior to leco-analysis for plant nitrogen content. Then the plant nitrogen values will be combined with yield adjustments to calculate two crop physiology NUE indices, the Nitrogen Nutrition Index and the Nitrogen Harvest Index. So the Nitrogen Nutrition Index is the concentration of tissue N under field conditions divided by the tissue N concentration under luxury or high fertilization conditions, whereby high Nitrogen Nutrition Index values that are close to 1 are interpreted as near optimal plant N status, where if the values are less than 1, it indicates deficiency. On the other hand, Nitrogen Harvest Index is how efficient the plant is at placing all of the acquired N into the seed, by partitioning N as uptake at maturity, plus the N transferred or remobilized from existing stem and leaf structures into the seed. So together, Nitrogen Nutrition Index and Nitrogen Harvest Index improve understanding of the uptake of N throughout the life cycle and movement within the plant into yield to maximize NUE. As mentioned, we performed an extensive field study on the diversity panel, and this took place in 2019 and 2020 at Saskatoon and Melfort Ag Canada sites. The lower field N treatment was set at 60 kilograms per hectare of available N, whereas the high or luxury treatment was 120 kilograms per hectare. The sampling, partitioning, and analyses for these trials are very similar to what was described for the agronomy study, just at a much larger scale because of the number of cultivars. The values that will be calculated from this trial are also very similar to the agronomy trial in that Nitrogen Harvest Index and Nitrogen Nutrition Index will be used to assess the natural variation in NUE across the historical panel. So pictured here are a couple snapshots of the trial in Saskatoon in 2019, and also is a picture of Rajiv Dekal, who is the PhD student who will be performing these calculations. In order to take a sneak peek at the trial results in 2019, we've taken an initial look at variation for yield from the Saskatoon and Melfort sites. What is shown on each of these charts is the yield on the x-axis compared to the coefficient of variation, or dispersion of yield points, on the y-axis. We did this expecting that the cultivars showing high CVs are more likely showing differential responses to the two nitrogen treatments. Using this approach, we were happy to see the expected trends, in that hybrids were yielding more than OPs across both sites. Looking at the range of CVs between groups, hybrids and OPs were similar in range, with greater range of CVs in the breeding lines, suggesting greater variation for nitrogen responsiveness exists within this group, which is a really good thing if one intends to study the genetics of NUE. A notable observation from this preliminary analysis was the range in CVs across the hybrids, supporting that hybrids may be responding to nitrogen differently. So to confirm this, a proper multi-location analysis on this data will need to be performed to see if there's consistent groups within the cultivars that are behaving similarly under different conditions. If so, this would be an indication that further optimization of G by E by M, or genotype by environment by nitrogen management, could lead to greater economic and/or environmental efficiencies given the range of hybrids we already have available commercially. Something that is very exciting that has come out of this NUE CAP project is the opportunity to partner with collaborators in the Plant Phenotyping and Imaging Research Center, or PERC, at the University of Saskatchewan. Through this partnership, we've been able to increase both the aerial and ground-based imaging capacity in the field trials for this project. Equally important is the PERC capacity for image processing using their user-friendly innovative pipeline called Plot Vision, and also the collaboration with computer science researchers. As we move into deciphering genetics and breeding questions around NUE, the digital phenotyping tools will be very critical for enabling the scale that is required. For this, Sarah Van Steenbergen, a PERC master's student, is looking at banana founder lines grown at low and high nitrogen treatments in Saskatoon to develop prediction methods for plant nitrogen content. To assess root system architecture in the field, we're using two systems. The first is the use of mini rhizotrons, which involves the insertion of a clear plastic tube used for imaging of the roots in situ as they grow down around the tube. The second method is using root pulling force, which measures the amount of force required to pull a plant from the soil. There's a group out of Colorado State University who have used this method extensively in brassicas, but we are assessing the method in our NUE trials for possible use in larger breeding trials. What we are finding is that root pulling force consistently ranks different cultivars between N treatments and seasons. However, it does not consistently show differences between the N fertilization treatments across the years. Melissa Arcan's study explores the relationship between root architecture traits, NUE, and soil nitrogen cycling across different cultivars under the contrasting soil conditions and fertilizer regimes. The hypothesis is that variation in nitrogen cycling processes among the different canola lines exists. And if this is so, then rhizosphere nutrient cycling might be governed by heritable plant traits associated with improved nitrogen uptake efficiency. And this could not only be studied further, but potentially selected for. So pictured on the right is Zhou Zhu, who's a master's student sampling a plot at the vegetative stage. Looking at the activity of a soil enzyme that's involved in N cycling, called N-acetylglucosaminidase, or NAG, there were a few cultivar-specific effects seen, and they depended on developmental stage at both Saskatoon and Melfort sites in 2019. So Joe and Melissa have also looked at NAG activity between the row compared to the rhizosphere, and these results have shown that the canola roots are clearly contributing to the NAG activity. In addition to looking at enzyme activity, abundance of bacterial ammonification genes have also been examined in the 2019 trial. So there's evidence of more bacterial action earlier in the field season at the bolting stage compared to mid-flowering. Also, there are more copies of the gene at low N rates, which is indicative of potential enzymatic activity for N cycling. So overall, the results to date show trends that suggest that low nitrogen fertilizers reveal greater differences between the cultivars for N cycling indicators, suggesting that nitrogen uptake efficiency differences might be better differentiated under limiting conditions. But the remaining analysis from 2019 and those from the 2020 field season will clarify this if this trend is robust. So the objective of Raju's study is to use nitrogen isotope discrimination as an integrated measure of nitrogen flux assimilation and allocation in plants. The hypothesis being tested is that 70 years of direct selection for yield has indirectly selected shoots and root traits that have contributed to genetic gains, and this trend is associated with nitrogen use efficiency. So the physiology framework is that plant nitrogen fluxes during the uptake of inorganic nitrogen from the substrate pool and assimilates either in the root or leaf organic sink pools. And the proportion of nitrogen assimilated in the leaves is the ratio between the organic and inorganic nitrogen translocated to the leaves. The setup of the steady state system is a hydroponic 2,000 liter N reservoir which contains different concentrations and forms of nitrogen, either nitrate or ammonium, along with Johnson solution for other macro and micronutrients. Yi Hu is a PhD student at UBC and is assessing the response of two forms of nitrogen at different concentrations. And in this setup, the growth tank houses 4 replications of 32 cultivars. Canola seeds were first germinated for 1 week prior to transferring to the hydroponic system. This slide is showing the low nitrate treatment for which the nutrient medium was completely replaced as necessary about once a week. The picture in the middle shows the first week of growth after transplanting. Plants were harvested when the earliest cultivar started to flower in order to exclude N remobilization and translocation for seed production, which would affect the model prediction. After harvest, each of the plants were separated into leaves, stems, and roots for biomass, as well as nitrogen isotope analysis. As part of the CAP NUE project and working with PERC, Isabel Parkin and Steve Robinson have had the opportunity to use the high-throughput phenotyping LemDetect Scanalyzer system at Nebraska Innovation Campus. Using the NAM founder lines grown at contrasting water treatments, digital signature for key traits were generated. A range of spectra were used to image the plants regularly. From this, Steve and Isabel have been working closely with Jordan Evans and Ian Stavnis from Computer Science to test latent space phenotyping, which is a deep learning approach to plant image analysis where different treatments have been applied. The work at the Nebraska facility will lay the groundwork for utilization of a system that is being installed at AFC in Saskatoon. The plant array system is an exciting phenotyping system that will be described in detail in a presentation tomorrow by Dr. Moshlian as part of Canola Innovation Day. In short, this system is designed to carefully manage water and/or nutrients each individual pot receives. It precisely measures key yield-related traits and continuously measures the shoot, root, and the environment. The system is designed specifically to study physiological processes. Thus, we are anticipating using it to help decipher NUE and canola. In summary, we are excited about our progress to date on this project and look forward to finishing up lab and statistical analyses over the next year. Without the outstanding support of field staff at both Saskatoon and Melfort Ag Canada locations, this project would not have been possible. There's also been an extensive team of biologist technicians and students from Ag Canada as well as the U of S helping with sampling and subsequent analysis, with special mention to Jana Ebersbach, who analyzed the high-throughput phenotyping image data. We were lucky to have the opportunity to move forward with field trials this past season, and doing so safely was enabled by administrative and management divisions from both of our institutions. I would like to quickly acknowledge industry in-kind contributions in the form of seed and irrigation water to this project, as well as contributions by collaborating organizations. Lastly, I want to thank the 3 provincial canola grower organizations for prioritizing this project for matching funds from the Canadian Agricultural Partnership, which is administered by the Canola Council under the Canola Cluster. And with that, I'd like to thank you for your attention, and I look forward to discussing our project with you either in the upcoming Q&A panel or after the conclusion of the afternoon session at a Canola Week virtual table in the networking lounge.
Good, thanks, Sally. All right, we've got a few minutes for questions, and then right after that, I'm doing this live polling session. So hang in for that right after this Q&A session. That'll be kind of fun. Anyway, Nate, you wanted to just note on something that was in your presentation that you wanted to clarify?
Yeah. So I'm now realizing that one of the downfalls about pre-recording a presentation is that if you make a mistake or unintentionally forget something in your presentation, you don't get the chance to fix it until now. So with that being said, I would like to address a key funding agency that I forgot to include as one of the key funders in the Canola Agronomic Research Program, or CARP, which is the Western Grains Research Foundation. We are excited that the WGRF is a part of the CARP funding framework, and we're looking forward to continuing to partner with them for the foreseeable future. So I apologize about my mistake during my presentation. And yeah, thanks, Jay.
Right on. Yep, good, Nate. Good to acknowledge WGRF. Right on. Okay, question for Dr. Ma. What is the reason or explanation for increases in yield found in canola when rotated with wheat? Did you measure the wheat effect? Yeah.
Okay, can you hear me?
Yeah.
Okay, I guess there are several factors contributed to the yield increase, but the first and foremost is better use of resources. That's because the wheat and canola have different root systems. So they will use different nutrients and the water. So when you have this kind of rotation, you will have a better balanced use of these resources. And also another possibility is the disease, biotic factors. Maybe that's also important. For this particular study, we have not quantified any of this, but in the first objective, our collaborator at the current first will look at the soil biome to see if we can link to identify the soil macro factors contribution.
And you also said that you haven't done the root or fully dug into the root part of your study. That'll be interesting to see how root length, size, and biomass will affect yield and nutrient uptake.
Yeah. For the root part, we are mainly interested in the comparison between pre-plant versus split application. And because that's a lot of work to be done on the root part, the work only done in Ottawa for the past 2 years, 2018 and 2019. Of course, this year we have very limited access to the field. So the results have not summarized yet.
So we may find out though, whether if you have all of the nitrogen there right at the beginning, do the roots get lazy? I've heard that term used with pulses, but maybe it could happen with canola too, where it's better to have a split application?
The idea for split application is number one, when we plant the canola, so there's a low demand. So when nitrogen is sitting in the field for 1.5 months, so there's not cannot be used right away, but have the risk for loss. And another one is when you split, you have the chance. For example, if you have a drought by mid of June, you will know the situation is, you will more justified to use the right amount.
Yeah.
In terms of if affect the root development, well, it's in general biology, if you have too much water, too much nutrient to start with, the root probably not to distribute the energy to explore large area to seek for the
which could run into could get a crop into trouble if it gets dry later in the season and the root biomass just isn't there.
That's a hypothesis. We are not tested that yet.
Yeah, right. Okay, I won't put you on the spot there. Okay, one more quick question for you, Dr. Ma. What hybrids were used for the N-rate study?
Yeah. Because we have a large experiment across many provinces, the hybrids used are different from the east Prairie, from the west. In the east, including Ontario, we use the Invigor 233P compared to 252. In the west, in Beaver Lodge and Olds, and also in Scott, we use the PV 581, and we're also using the 6074.
Okay.
Basically, we wanted to see at the local level what's the most popular varieties we compare with.
Good. Thanks, Dr. Ma. All right, Sally, quick question for you, and then I'll move on to the polling. And then just so as a reminder, if you have more questions for the three speakers, make sure to join the virtual tables in the networking lounge later. But Sally, I think it's obvious from your presentation that this nested association mapping, this NAM work is incredibly complicated. But could you just give me like a 1-minute Coles notes of what we could achieve? I think it's how we can identify very complex traits and start selecting for those in canola breeding. That's my really rudimentary understanding, but can you clarify it for me?
Mm-hmm. Yeah, and again, you know, a lot of the discussion we've had with the team as a team about this is how do we address this complex question? And really what came down as we were laying things out is that anecdotally, we know that there's different plant architectures that we see in the field that are somewhat consistent or really consistent between environments. And you know, we knew from other literature, from smaller studies, that these plant architectural differences could be an indicator of different nitrogen use patterns in terms of the uptake and remobilization. So the strategy was to start with some really good field tests of bigger collections of germplasm. And so we have, you know, a stratified test of 2 sites, 2 years as a starting point to look at where the big differences are, and then to look at the nitrogen use efficiencies of those different lines as a starting point. But really, what I've described today, it really is just a starting point.
Yeah.
You know, and we're really just at the tip of the iceberg in terms of starting to look, going from the historical panel going into the diversity collections to see what's there.
Yeah, it'll be really exciting to see what kind of things this work can uncover when it comes to canola plant physiology, the phenotyping work, and we look forward to everything that you continue to do, both of you. All right, let's wrap up this part of it, and then so what I'm going to do is I'm going to start showing my screen here, and just going to click on. So bear with me for a second. So we're using a program called Menti. And I am going to do this now. Okay, so you should have something that says what best describes you up on your screen as you watch the presentation. And so at the top, this is how I want you to participate. Okay, somebody's already figured it out. Menti, go to menti.com, and then the code for our poll here is 4670909. It's right there on the screen. So I'll wait a minute or so. Many of you are maybe farmers and non-farming stakeholders. So I want you to pick one or the other. I'll leave it up to you to decide what hat you have on today. But I want everybody to choose, and we're up to 40. So I'll leave it for another minute. It'd be nice to get into the triple digits. I know we have somewhere between 300 and 600 people participating in the conference. My computer is working hard. If you can hear the fan going, I guess this is, this will be interesting. So we've got about 20 minutes to get this wrapped up, but there's only 14 questions, so we should be able to click along fairly quickly. Okay, so we've got 20 farmers and 108 and counting non-farming stakeholders, which is what I'm calling everybody else. So if you're involved in the conference, you should be able to participate. Okay. All right, so what best describes you? So we've got 22 farmers, 123 not. So we got 125. Oh, we got 146. So it's not everyone is answering, or we've got, oh no, it's pretty quick. Okay, we're gonna go to the next question, which is, so the next slide. Okay, so we're like, as the theme for the day is, we're talking about 4R. So just as a quick reminder, this isn't a question. 4R fertilizer management is right source at the right rate, right time, right place. Just in case you needed a little refresher on what 4R was. Okay. Oh, this is, boy, some of you are so quick. Excellent. Yes, you'll be able to click along on your own speed, but this is what's, I think it's called a cloud or a word cloud or something. So what's the first word or short phrase that comes to mind when you think of 4R? So the more people click sustainability, the more emphasis it'll get. So you can see sustainability is the top one so far. Although as people come in, it's losing a bit of ground. We got efficiency, we got fertilizer, we got stewardship, we got rate. Oh, this is fun. And I've got, I think Warren, sorry, you know what? I've moved my camera, so now you can't really see me. This is really awkward, but I've got this up on my back screen. So if you're wondering what in the world's going on, I'm going to rearrange things a little bit. I just checked my image and you can't see me. There we go. Okay, so I'm actually looking up at this big screen, which is why I'm not making eye contact with you. So I apologize for that. All right, so we're going to go on to the next one, but this is, you can see how there's lots of great input there. Common sense, I like that one. What else do we have? Doing what is right, improvement, optimal productivity, good stuff. Okay, do you think 4R is a valuable initiative? So we've got, I think that's a combination. Yeah, so 86% of non-farming stakeholders. Oh, no, that's not, I'm reading this wrong. So the total is basically 100% except for 2 people who think it's a valuable initiative. I don't know who anybody is, so you can feel free to say whatever you want. I don't know who anyone is. This is completely anonymous. So the 2 brave souls who said it's not valuable, good. It's good to have different perspectives. Okay, there you go. So pretty clear that people think it's a valuable initiative. All the farmers said it was, so that's good. What is the biggest barrier to 4R adoption? Okay, good. So this is, you're not going to be able to read this, so I'm going to read it to you. But when you're answering the question, you can see pretty clearly what it is. But what's the number 1 spot there is education. So the benefits are clear, but we need to show them. The farthest bar on the left, the one with a 35 on top right now, is cost. I can't see a return on investment or time. And then logistics, too complicated. So these 3 bars are education number 1, logistics number 2, and then right behind there is cost. And you can see the blue bars are the farmers. So the farmers are fairly, well, everyone's distributed across all 6, so everyone has a thought there. Unfortunately, I don't know what other is, but maybe if you have strong thoughts on other, we could chat about it during the networking later.
And Jay, it's Warren Ward here.
Hey.
I'm keeping an eye on the comments here in case anything does pop up, but nothing so far.
Okay,
interesting though that on the cost angle, where we've seen in the past that that's a limitation for some people adopting 4R practices, but it's also what's driven other people to adopt these 4R practices. That's an interesting one.
Yeah, good. So it's kind of not surprising that cost is popping up there, but like you said, Warren, for a lot of people, saving money or improving your fertilizer efficiency is the main reason to do 4R. So there's often those, it's cost or input. Yeah, input cost shows up on both ends of the spectrum. Good. All right, how often, okay, this is about soil testing. So of course, everybody knows that soil testing is a pretty important part of the 4R process. It's right there at the basic level that there's the nutrient management decisions are based on some sort of soil testing. So how often should a person soil test each field? Overwhelming answer is every year. Every 2 years, actually funny, every 3 years has a slight edge on every 2 years, which is kind of interesting. All right, now the next one, and a lot of people probably jump forward to it, but that's no problem. So we go from how often should a person soil test to how often does it actually happen? So farmers will know, obviously, how often they soil test. For everybody else, it may be a bit of a guess, although it will be an educated guess for agronomists and people on that side of the business. So every 3 years seems to be the leader right now, including among farmers. So there you go. So you know what, so the farmers and non-farming stakeholders were in sync there. Kind of interesting to see how the numbers are in good alignment. rarely or never. That's higher than I thought it might be, but still it's in fourth place, which is good. And then that every 2 year, it's two slides in a row. The every 2 years choice was down there pretty low. So it's either every year or every 3 years seems to be the common answer. Warren, anything popping up there in terms of questions or comments?
No, so far I think everyone must be just following along on their Menti app, Jay.
Yeah, it's fun when you get to participate actively in an event. So I'm enjoying this. Thanks everybody for participating. All right, is there enough soil okay, so this is a question on if everybody decides to soil test, can we get all those samples tested? So is there enough soil testing capacity if everyone chooses to test fields in 2021? So 97, 98 votes for no, 69 votes for yes. So a slight edge to no. Some might call that more than a slight edge, but whatever.
Jay, on this one, do you know if the colors are representing the farmers and non-farmers? We do have quite a split there.
Oh, you know what? That can't be. Oh, sorry. Good. Yes, good, Warren. I didn't split every question, and I didn't split this one. So this one, yeah, good point. So this is everybody's lumped in here together. All right, this one's back to the split. It's kind of a neat feature of Menti is that you can decide which questions you want to split versus not. Okay, so lots of choices here. And if you're still answering this question, you can pick more than one answer. So if you use fall application and seed placed and in crop top up, you can answer all three or click all three boxes. What do you think, Warren? Any surprises with this one?
Jay, I was just wondering if you could read out the categories there. They are a little bit.
Yeah, of course. Yeah, I'll do that for you, Warren. All right, so the big one, the one second from the right, time of seeding. So the best or the preferred way to apply nitrogen is at the time of seeding. That's the leader. Second place is spring band pre-seed, and then spring broadcast incorporated. So the big three are all in the spring. And then we got fall broadcast incorporated.
Yeah, so timing wise, I mean, that is encouraging. And it's not surprising that at seeding is leading the charges.
Yeah. And even so, look at the interesting one maybe is the fall band. So there's lots of farmers who do that. But if you think about our 4-hour discussion from this morning, Warren, how we were encouraging the banding, or not we, but I mean the panelists were encouraging the fall band or a banding option. So the people who are applying in the fall, or the farmers who are applying in the fall, banding is a pretty popular choice, which is good to see. Just for fun, so the broadcast without incorporation, very low, and only one farmer does fall broadcast without incorporation. So that's not, I would say that's outside 4R, unless your fall broadcast, what the choice is, and maybe this person who chose this fall broadcast is using an enhanced efficiency product to prevent losses. So that would be the one case where a fall broadcast was 4R, is that if you used some sort of enhancement product to reduce losses. So great stuff. Where do you see variable rate fertilizer adoption in 5 years' time? So as we move from basic to intermediate to advanced 4R practices, this notion of looking at zones and applying based on productivity ratings for zones within fields, that's going to be something that we move toward as an industry. So I was kind of curious with this question as to what people thought of this. So less popular, not very many people think it will become less popular. Status quo, so it'll stay about the same. There's a few there, but more popular is the overwhelming choice here. And then the next choice, I missed a space there. So that actually says a common practice on most farms. So is it more popular, or has it gone from more popular into common practice? So that's what I was wondering. So there's a bit of a distinction there. But yeah, so that's good to see that there's a definite openness to looking at that. People see the benefit in variable rate. It's just like we talked with the farmers in the panel this morning. Sometimes it's a cost factor, logistics. Changing your seeding tool is not a simple, easy decision. So maybe there's ways that we can think of to make this a little easier for people to consider variable rate. All right, this isn't the best way to ask this question, because this will be many pages. So this will be good for me to go back afterward and read all these answers. The cloud is a little better, and we'll get to more clouds in a bit. But so this is, what practice do you think will make the biggest difference in terms of improving fertilizer efficiency or nutrient use efficiency? So there's lots of good tips there. The ones that I can see, I guess, and that you can see are, well, placement pops up quite a bit. Site-based, site-specific, I'm assuming that is. Right hybrids for our education, economics, logistics. Yeah, right. Runs the gamut there of a lot of the big factors. Okay, so this one I'll have to go back through afterwards, and maybe we could do a bit of a summary in Canola Watch next week. canolawatch.org. If you're not a subscriber, you could find it there end of next week. We must be getting close to the end. What research or agronomy? And I've covered up my clock on my phone, so we're at 38, and I think I have till 45. So we've got 7 minutes, so this should be okay. All right, this is the discovery part of the day, and we want to make sure we poll the audience when we do each of these canola discovery forums, just to see what people are thinking. What's missing to help us improve fertilizer efficiency? And I suggested 1 to 3 words just because it's easier when these clouds pop up. Don't know is the #1 answer so far, and that's fair enough. I mean, this sometimes is a bit of a mystery to know where to go next with some of this research. And I think to the point with Bao-Luo and Sally Vail's presentation that we just all watched, there's some really exciting things happening, and maybe we don't know how it's all going to shake out, but it's pretty exciting to see how we might be able to improve the plant itself to enhance nutrient use efficiency. Education. So the big ones now are don't know and education. You can all see that. Soil sampling. Soil sampling is missing. Okay, so we got don't know with an apostrophe and don't know without an apostrophe, but they're both big ones.
Quite a bit of not knowing there, Jay.
Yeah.
Interesting soil sampling entering the mix there, and that would really drive towards that right rate decision as well. Which, if we think back to the panel from this morning, that was one of Mario's #1 things that we would need to look at from a research perspective. Yeah, that's good, Warren. Anything else popping up there that you see? Foo dust dismissal. I don't know why that one popped. It's out of all these little ones, that's one that caught my eye. I see logistics in there as well, and we always see that one as being an issue when it comes to fertility management, right? Is logistics do play a big role in it. So that making those changes over time, and that's the nice thing about the 4R practices is that we can't always do the right thing all the time, but it is a learning process where if we have to give a little bit here, we can take a little bit there and try and maximize our or reduce the impact maybe of some of the practices that we don't like. Yeah, great comments, Warren. It's good to have you here. Otherwise, it's just me talking all the time. Okay, I think we've got maybe 1 or 2 more. Who is best equipped to deliver fertilizer tips and advice
Canadian canola farmers. So if there is a gap in education, or if or at least there's some headway to be made, how do we close that gap? Who are the best people to share that information? And agronomists are topping the list right now. Canola organizations, including CC Agronomy Specialists, is right in there behind. That would be my colleagues. They're a small group, but boy, they're effective. Warren being one of them, and getting the word out on unbiased canola agronomy information, research-based information, and that's why. I mean, this is why this canola event is such a big part of our year, is that we get a chance to get updated on what's really happening, and then get a chance like this to get some input from everybody. So if I didn't say it enough, I really appreciate you participating. Thank you. Okay, good. So agronomists and canola organizations, including CC Agronomy Specialists, are running away with this one. Good to see. All right. Okay, this is the very last question. Just to wrap up the 4R theme. What do you think it will take to get 90% of growers using 4R practices? Again, this is the cloud, so 1 to 3 words is what I was hoping for. Education, clearly the bold one in this case. Incentives are popping up there. That's an incentive and incentives. Yeah, economic
so it seems
incentive.
Like
yeah, go ahead.
A bit of incentive there, Jay. Interestingly, you know, if you wanted to look at incentives for adopting 4R practices, the return on investment and just the better practice on these better practices, in some cases, I think a lot of people who have gone through the 4R program are seeing that the incentives are there without having somebody external in the industry providing that incentive. And I interestingly do see your name popping up there as well, Jay.
Well, I appreciate that. That's interesting. Well, there's 10 of my work colleagues, so if they all put my name in there, it would pop up like that. Anyway, that's it. Let's wrap up. Again, this is really good. We're going to save all this information. If you think of someone else who you think would be good to take a stab at this, you feel free to share menti.com and then this code. I'll wait 30 seconds if you want to jot it down. And then it is open to anybody. So if you want to share it, I suppose we could share it on Twitter, I suppose, although maybe I should save it and restart it if we do Twitter. But anyway, yes, we can share this around so we go beyond our small group here. All right, I'm going to wrap up. I'm going to stop sharing my screen so we can go back to the program. And that's Warren's popping up. Thanks, everybody.
Hello, good afternoon, everyone. Thanks, Jay, for hosting and moderating that last session. My name's Andrew Sharp from the Global Institute for Food Security, and I'm the host and moderator for the next session, which is soil health and novel research. I'd just like to point out, if you do have a question, please make use of the button, the Q&A button at the bottom of the screen. And it would be great if you could indicate which speaker your question is for when you ask the question. The first talk is from Ben Ellert, a research scientist at Agriculture and Agri-Food Canada in Lethbridge. And his talk is entitled Climate Change and the Soil Microbiome.
Hello, my name is Ben Ellert, and I want to present to you a biogeochemical perspective on greenhouse gas emissions. What is biogeochemistry? Biogeochemistry refers to the transfer and transformation of chemical elements amongst major components of the Earth system or ecosphere. Those components being the atmosphere, the biosphere, which is all the biomass on the planet. The biggest portion of the biomass consists of plant material. Material. The second largest component is the microorganisms, the archaea, bacteria, fungi, and the like. Us animals account for a smaller but important part of that. The lithosphere is the Earth's crust, the domain of the geologists. The hydrosphere, of course, everything is bathed in water. And the fifth sphere you may be less familiar with, that's the noosphere, or the influence of the human mind on all these processes. Biogeochemistry is inherently interdisciplinary, as is soil science, and the intersection of all of these components is the soil. The soil is teeming with life. It often has mineral components, part of the lithosphere. Of course, there's water, essential for plant production, and there's pores exchanging gas with the atmosphere, strongly influenced by humans. So it's the ultimate interface, and we mismanage it at our peril. The founder of biogeochemistry was Vernadsky, a student of Dokuchaev, early soil scientist, as well as the famous chemist Dmitry Mendeleev. Already in 1926, in his book, Vernadsky recognized that humans were becoming a mighty and ever-growing geological force. So what does this geological force look like? Well, one of the things we can look at is the iconic Keeling curve showing the increase in atmospheric CO2. This is monthly data for the Mauna Loa Observatory in Hawaii and the data from the Earth Systems Research Global Monitoring Lab in Boulder. The increase in CO2, early on, it was attributable mainly to land use change, human activities. 500 years ago, transferred CO2 from the Earth's surface to the atmosphere as a result of deforestation, clearing of land for agriculture, and that sort of thing. In the past 100, 200 years, we've become increasingly reliant on fossil fuels, and digging fossil fuels out of the ground and burning them also is a major contributor to increasing atmospheric CO2. This sawtooth pattern reflects seasonality because these are monthly data. So in the summertime, when plant growth is the highest, the plants tend to pull CO2 out of the atmosphere to a minimum point. But each summer, that minimum point becomes higher and higher because of continual release of fossil fuels to the atmosphere. Now I want to zoom in and take a little bit closer look at this. One of the tools that biogeochemists use are isotopes, and isotopes are chemical elements that have a very subtly different atomic structure. Carbon-13 is much like carbon-12. It's a stable isotope, but carbon-13 has one extra neutron, and it's useful to track the flow of elements in ecosystems, and in fact, the entire ecosphere. So we see the blue line shows the increase in atmospheric CO2. At the same time, the amount of C13, the relative amount of C13 in that is decreasing. Why is that so? It is so because fossil fuels contain relatively low amounts of C13. The more fossil fuels you burn, and what happens is you cause a decrease in the C13 levels in atmospheric CO2. So what that is that's an isotopic fingerprint, conclusive evidence for the influence of humans, the noosphere, on the ecosphere. So the ecosphere is the total global ecosystem. Here we want to zero in on crop system in Western Canada. This shows the cycling exchange of carbon between cropland containing 70 metric tons of carbon per hectare in 0 to 30 centimeter layer, This system, we have a net carbon accumulation in plant matter of about 4.5 metric tons of carbon per hectare per year. A portion of this is harvested and exported. Maybe China wants to buy that, maybe. The residues go into the soil, but they don't just accumulate there. Typically, around 80% of that residue carbon will be decomposed within 2 years of falling on the soil. And in this particular situation, we have exact balance, inputs equal outputs, so that carbon stock will persist. Any imbalance, either way, it may change that carbon stock. Now I want to take a closer look at carbon stewardship because that influences CO2 emissions from our cropping systems. So the analogous to the 4R nitrogen fertilizer stewardship, the first thing I want to look at is rate. What determines rate of carbon input? Simply depends on yield. That's the primary. How much plant growth do you have, more carbon input. Another important factor is the plant carbon partitioning, the distribution of that carbon amongst the harvestable parts, like the grain, versus the residues. And here, the above ground residues are in green, and the below ground residues, which consist of the roots and root exudates, are in red. And these are for common crops. Very nice review of that in the recent Canadian Journal of Soil Science paper. They also have data on the nitrogen content, very useful data set here. The thing that sticks out is canola is very productive. This is pretty high yielding canola. And the other thing is the fraction that is harvestable and exported is relatively small, so that leaves a lot of residues that go back into the soil, and that has important implications for both CO2 emissions and N2O emissions. The next are timing of canola residues, the right time. Well, the timing of residue inputs simply is determined by when it's ejected from the combine. Hopefully that's in the fall of the year and not delayed till the springtime after it's overwintered outside in the snow. The other thing that we need to consider is the position of canola in a multi-year cropping sequence or crop rotation. If you have more than about 1/3 of your land under canola, you're aiming for a pest problem, I believe. Canola residue placement, right place. Where do we put the canola? Usually it's spread on the soil surface. Standing stubble tends to be drier, so there's less biodegradation, but it's more exposed to weathering, so there's a bit of trade-off there. Below ground residues of canola, the rooting system, most of those are allocated closer to the soil surface than other crops, so that makes it more susceptible to decay. Tillage, generally we try to keep tillage out of our cropping systems. It buries residues and greatly accelerates decay. Residues sometimes are harvested in the irrigation districts in southern Alberta, especially if it's a drought year. You have heavy canola crops, lots of residues. Those residues will be harvested, and I know a lot of livestock producers find it useful as bedding. But there's also interest in canola residues as a biofuel. Now, this isn't biodiesel part, which has received lots of buzz. It's cellulosic biofuel. And in the inventory mapping tool, they don't explicitly report canola residues like they do for other cereal crops. But in this paper, they do explicitly map out the potential contribution of canola residues for cellulosic biofuels and or bioproducts. Now the final R, the residue form and in the plant material and how that chemical form is transformed during decomposition. This shows the plant cell wall, increasing magnification of the chemistry of the plant residues. Key feature of all terrestrial plants is this, at least the vascular ones, is this fibrous structure, and that consists of cellulose. Cellulose is a crystalline polymer consisting of glucose molecules. It's the most abundant polymer on Earth. The most abundant phenolic polymer on Earth is lignin. And cellulose and lignin are intimately associated with each other. They're glued together by hemicellulose, which is an amorphous polymer of carbohydrates, which is less resistant to extraction and what have you than cellulose. There's a number of ways we can look at this. The biorefinery people look at it one way. The people interested in this lignocellulosic material as a feedstuff look at it another way. This compares straws from common different crops. These authors weren't very enthusiastic about canola straw as a feedstuff, although I know some people do feed it. But the bottom line is there's not a lot of difference in the plant chemistry when it comes to the detrital food web. Our soil microbes can find a way to decay pretty much any plant material you throw at it. And there's lots of interest in how that's transformed into soil. What determines the extent to which those residues are retained in the soil and what is released back to the atmosphere as CO2. Early on, the humic acid theory was based on analyses where they extracted the soil organic matter with strong alkaline solutions, and they looked at the structural chemistry of the organic molecules, and they thought that these large chemical structures were resistant and retained in the soil. Other soil scientists says, no, it's not the chemical structure of humic acids, it's actually the physical location of that organic material in association with the clay particles and silt particles. And it might not even be plant material anymore, it might be the microbial tissues that were produced by feeding on that plant tissue. So there's lots of controversy on that. How do we measure CO2 emissions? Well, there's a number of ways of doing that. The most straightforward way is simply measuring the change in soil carbon stocks. You can't expect a change to occur in less than 5 years. 5 years is the absolute minimum. It's best to measure change, say, between a temporal span of about 10 years, because the inputs and outputs are small relative to the stock that's there. We use a coring apparatus. We take large volumetric cores with careful measurements of bulk density, carbon concentrations measured on those same cores. So to quantify soil organic carbon stocks ultimately will give you an indication of CO2 emissions. I get a lot of questions on that, so I just wanted to go through a few key points that often are points of confusion. On your soil tests, you often get reports of soil organic matter. Soil organic carbon is not equivalent to soil organic matter. Soil organic matter cannot be determined with adequate precision. What you should be measuring is soil organic carbon, and that's analyzed after you eliminate the carbonates. That's analyzed by sophisticated automated elemental analyzers. And once you get those concentration data, the soil organic carbon concentration is not equivalent to the stock. The soil carbon stock places the lab data into the field context based on horizonation, profile morphology, bulk density, etc. And when you compare the carbon stocks at 2 points in time to determine whether you're accumulating carbon or losing soil carbon, the soil organic carbon stocks must be based on equivalent soil masses. So you have to adjust the soil thicknesses so the masses are the same. And these soil organic carbon stocks reflect the balance between plant C inputs 2 and CO2 outputs from the soil. These eventually and inevitably will re-equilibrate. Now I want to move on and talk a little bit about N2O emissions. This is a field with some nitrous oxide flux chambers. That's a close-up view of the chamber for fertilizer bands. We also use different shaped ones. Unfortunately, we cannot measure a stock and infer N2O emissions from that. We have no alternative but to actually measure the fluxes of that trace gas from the soil. And we do that by collecting gas samples and measuring them offline in this case. A few comments on soil N2O emissions from canola system. They're highly variable in time and space, presents analytical challenges. There's default N2O leaks from fertilizer use. The default leakage value is about 1 kilogram of N2O nitrogen per 100 kilograms of fertilizer N applied. Fortunately, on the prairies, our leakage from fertilizer N is a little bit less than that, maybe 0.8 kilograms from the black soils, decreasing all the way down to 0.016 kilograms of N2O per 100 kilograms of fertilizer use. So not a big leak. It's not going to make a farmer lose much sleep about that nitrogen loss from an economic perspective, but environmentally, it's a bad thing because N2O is a potent greenhouse gas. Atmospheric N2O concentrations are increasing. And it also catalyzes stratospheric ozone destruction, so a double environmental whammy. These measurements refer to direct soil N2O emissions measured using flux chambers. Indirect emissions are rarely measured, but they're important. So that's measurements after you have leakage of reactive N to waterways and the atmosphere and such. And N2O leakage by any means is an important determinant of environmental performance of the system. This is a chart showing some fluxes from Scott, Saskatchewan, a very nice paper by Reynold Lemke. This is, the arrow indicates when the fertilizer applied. We see the spring burst of N2O emissions and then bigger emissions during the growing season. The top panel is for wheat following canola. The lower panel is for wheat following wheat. We have more N2O emissions on the wheat that's following with that big dump of canola residues the previous fall. So we have to pay attention to that part. To mitigate soil N2O emissions, the starting point is standard for our fertilizer and stewardship, but we have to move beyond this. We've got microbial transformations of crop residues. They determine both CO2 and N2O, but they also influence oxygen and dinitrogen, N2 concentrations. We might consider dinitrogen losses. They may be an expensive inefficiency, but emissions of dinitrogen may be preferable to N2O. In addition to fertilizer N, we have to manage the entire cropping system, not just canola. We have to pay attention to the residues, as Reynolds' data suggested, and livestock manure, reactive N leakage to the atmosphere. Final thoughts: carbon and nitrogen cycles are intertwined. Optimal environmental performance is critical in this age of increasing scrutiny and ever increasing greenhouse gases in the atmosphere, including N2O. The relatively small fraction harvested as oilseed means there is a considerable amount of carbon and nitrogen to manage in canola residues. For our N stewardship is an excellent starting point, but we also need to move beyond this. Things like substances like enhanced efficiency nitrogen fertilizers are not a silver bullet, and they are not an adequate substitute for knowledge and understanding of things like synchronization of soil and supply and plant and demand. And with that, I'd like to thank you for your attention. And there's my contact information. Thanks again.
Thank you. Thank you for that talk. Unfortunately, I can't start my video at the moment, so I'll just introduce verbally only. So the second talk is from John Sulik, is an assistant professor at the University of Guelph, and his talk is going to be with respect to canola spectral remote sensing.
Hi, my name is John Sulik. I'm an assistant professor in the Department of Plant Agriculture at the University of Guelph. And basically what we're going to talk about today is how flowers impact remotely sensed vegetation indices, how we can describe spatial variation in flower density, how that can also be related to variation in yield potential. And then we'll discuss how we can use indices to automatically map transitions in flowering. Although there is a pure yellow color, and we can't even have sensors measure light in this portion of the spectrum, canola flower spectrum, canola flower canola flowers aren't actually pure yellow. They are an additive combination or an additive mixture of green and red light. And that's because the flower petals absorb blue light and green and red are what's left over. Those mix together to form yellow. So the basic way that a vegetation index works is it looks at a difference in the amount of light that is reflected from a surface. And typically it will look at the difference between two wave bands. And those wave bands could be near infrared and red light. And typically, as plants are healthier or more vigorous, they will reflect more near infrared light and reflect less visible light, such as red light or green light. And so we can see for this healthy plant, there would be a large difference between near infrared and red, 0.4. Whereas for a less healthy plant, there would be a smaller difference, so, such as 0.1. And a vegetation index fundamentally does just this. It looks for a contrast between two wave bands where you would expect sometimes opposing behavior or else one band to have variation and the other not to have variation with respect to some biophysical attribute of interest. For instance, the leaf area index, or how many layers of leaves there are per unit ground area. Now, this is tricky once you start to introduce additional canopy elements besides leaves. So NDVI is really meant to be sensitive to leaves occurring on a soil background. But if you add an additional canopy component, such as flowers, what those will do is increase red light reflectance. And as far as any vegetation index is concerned, it doesn't know how many layers of the canopy there are or whether a canopy element is vegetative growth or reproductive growth. So if you have yellow flowers, those yellows are reflecting additional green and red light. For the case of something like NDVI, additional red light. So NDVI would be confounded into rating or scoring a healthy plant as unhealthy just because flowers are there. But we all know that if a plant's flowering, that doesn't mean it's unhealthy, but that would throw off an index such as a difference index or the normalized difference vegetation index. And so this problem's been out there for at least 10 years that researchers have known that spatial variation in flower cover or flower density can confound the relationship between a vegetation index such as NDVI and a biophysical estimate such as leaf area index. But that still begs the question of how can we describe the variation in flowers themselves, right? Because we could have variation in some leaf attribute such as LAI, but that's not telling us and it can be confused by the variation in flowers, but how we directly measure that flower variation. One way to do that that we came up with was just basically by looking at a difference between green and blue light. And you can implement that as a normalized difference index where you just take the difference between green and blue and you divide it by the sum of the two. That's one way that you can do that. And that's because the flowers are reflecting green light and absorbing blue light. So that's your contrast. Other indices such as NDVI, this is a slightly more complicated formula than I showed you on the previous slide, but it's just, it's basically looking at a contrast between near infrared and red light. Another index that I'm going to show you is, and discuss in some of these slides, is called VARI, Visible Atmospherically Resistant Index. And that's mostly a contrast between green and red light. And it also does subtract some blue light to account for atmospheric effects. And it's interesting because these two indices, NDYI and VARI, VARI, you can calculate both of these using any camera that has red, green, and blue pixels, or else red, green, and blue monochrome imagers. And so we published a journal article. This is the first one that we published on canola. And in this article, we just talk about some basics of if you want to describe spatial variation in flower density or leaf area index during flowering, what are your best options? And so in this graph, it's showing you that basically a ratio of green to blue light or a normalized difference, you can relate that to flower counts. So if you look at each of these red squares, these are all plots of canola. and they were blooming at the time of this image. There's a picture from ground level that shows you the canopy condition. And what I did is I went in with a quadrat that was a half square meter, and I counted how many flowers there were in each one of these. And you can see on the y-axis the range of flower density per half square meter. So from about 8 to just over 600 flowers. What you can do is basically regress that count of flowers onto that ratio of green to blue light, just averaged for all the pixels in that quadrat, and you can get a linear relationship. I've got two separate data sets I did this for. One where I non-destructively counted the flowers, which it's hard to keep track of, and another one where we actually pulled all the flowers off and went and counted them in the lab later. Now, if you know how many flowers there are per unit area, then you also have some idea of how many pods there are going to be and eventually how much seed there's going to be. So we went on to publish another article where we talk about spectral considerations for yield modeling. And in that, we basically showed that that ratio of near infrared to blue light or the ratio of green to blue light, either of those are a suitable index to use to estimate variation in yield potential during flowering. Now, something else that I did along the same time that we didn't include in this publication, but that is interesting, is we compared a very low-cost camera to a very expensive multispectral camera that costs over $10,000. This camera cost about $30. And both of those have green and blue wave bands. Now with the single imager, we call it single imager because it only has one lens, whereas the multi-imager has multiple lenses to sense the different wave bands. This is just showing you what the maps look like from each of those. These were captured at maybe a couple seconds apart from each other. But what we did is we took that ratio of green and blue light and regressed the yield values onto those index values for each plot. And it's really interesting because the expensive camera is called a tetracam. The cheaper one's called a Canon. And if we look at the normalized difference yellowness index values for the tetracam and the Canon, we'll see that they look very similar to each other. And they even have very similar R-squared values. The strength of the relationship's very similar. And this just goes to show you, you don't need a super expensive camera to map spatial variation and flowering within your field. Now, these images were captured during flowering. And you'll notice that this is an NDVI map. And it shows you the NDVI's strength of a relationship with yield, which is very low compared to the basically normalized difference yellowness index. And when you look at how these values of either index vary across time, it's very interesting. So this is a satellite image of a canola field, and it shows it where it's bare ground to where it was at rosette and bolting. Here's an image of it flowering, and then after flowering during pod fill and maturity. And you'll notice that this NDYI, which is the green curve, it reaches its maximum value when you're at peak bloom. You'll also notice that NDVI actually has a local minimum at peak bloom. And so if you want to know when flowering starts in canola using remotely sensed indices, you can just look for when the difference, you could look at any one time point and the time point before then. For NDYI, if the difference between a time point and the preceding time point is positive, but for NDVI, the difference between a time point and the preceding time point is negative, then you've got a very strong indication that flowering has started. And if you were to look at a difference between two time points of, let's just look at NDYI alone, these were images that I captured with a Canon camera. And you can see some variation in flowering between these two time points, about 9 days apart. One of the things to notice here, in this area, the flowering was not happening. It was very weak. Whereas in this time point, it got much more pronounced. Whereas in this plot right above it, it was strong, and then the flowering ended. And so if we look at just taking this NDYI and subtracting it from one point in time to the next, we can get a map that shows variation in where flowering has increased, which would be these blue positive values, and where it's decreased, these negative red values. So we have a new article that just was accepted for publication in Agrosystems, Geosciences, and Environment about automatic detection of flowering transitions. And one of the take-home points from this, both of these are box plots from two different data sets. The box plots on top here are from some satellite imagery. And what they are showing is delta NDYI values. So I just showed you a map that showed delta NDYI, how areas are either increasing or decreasing. And this shows that if you look at a 16-day interval between observations, the difference in NDYI for pixels that are flowering is going to be positive, whereas pixels that are NDVI or VARI, either of those structural indices, those will be negative. Whereas you'll find the converse when flower shedding initiates. The structural indices will have positive differences, whereas NDYI will have negative differences. And you'll notice that the distribution of all these values basically has a threshold of around 0 that you could use to differentiate between flowering and shedding and the sign of the change of these index values. Now for a data set looking at 8-day, a whole entirely different data set, not satellite ground sampled measurements that we made, the same pattern holds. You'll have positive NDYI values as you're approaching the maximum bloom, and after maximum bloom, you'll have negative NDYI values, right? And so these patterns are consistent across data sets that we found. Something to keep in mind is this doesn't work out perfectly. If you do have an area that has a strong peak bloom, you will notice this kind of VARI will be this green index and NDYI is this yellow one. You'll notice they have a pretty much a contrasting pattern where one increases, the other's decreasing. However, you get a canopy with very light bloom like this one, you can use NDYI to determine when you hit peak bloom. But using NDVI or VARI to indicate when flowering begins or ends isn't very practical because NDVI isn't sensitive enough to the presence of the flowers. And so if you were to apply this algorithm using that 0 threshold and just the difference between NDYI and a structural index such as VARI, you can map out these flowering transitions across space. So here we look at the difference between 10 July and 24 June. Areas that started to flower between these two time points will all be flagged in orange in this map. Same thing goes between these two time points. Between 26 July and 10 July, anything that started to flower, such as this field right here, right, that would be orange, whereas areas that stopped flowering would be in purple, right? So this field right here, it stopped flowering, and then that goes in purple. And you'll see that general correspondence. And then anything that stopped flowering between these two time points is going to be purple in this map. And so this just goes to show how you can use these general principles about the relationship between a reproductive index such as NDYI and a structural index such as VARI, and some simple rules to automatically map when those transitions occur. Or you can just use NDYI by itself, as I showed a few slides ago, to show just within a plot how flowering was increasing or decreasing in a certain section of it between two time points. And so hopefully this gives you some ideas for how you can use these techniques. Different applications for looking at relative differences in the field and yield potential, creating management zones for things such as swathing the canola to ensure uniform harvestability, and statistical yield models, and maybe even use this for determining binary fungicide spray applications. So with that, I'll conclude my talk, and thanks for your time.
Okay, thanks very much, John, for that talk. I think we have some time for some questions, hopefully answers from both Ben and John. I think they're both available in the Zoom setup here. So I see Ben's there. Hi, Ben. I got a question to get things rolling. With respect to the soil core sampling that you had in the slide there, how deep do you actually go for that sampling process?
Well, the deeper the better. I like to say 60 centimeters is sort of a minimum, especially if you have any cropping system with a perennial crop in your crop sequence. You want to be able to capture those subsurface changes as well. So that's ideal. Anything less than 30 centimeters it's not really relevant to be talking about soil carbon stock, I would surmise. So 30 to 60 and deeper if you've got a lot of perennial crops.
I suppose that depends a bit on the type of soil that you're actually sampling, perhaps.
Yeah, and the rooting depth of the crop. What do you, if you're trying to assess changes in soil carbon storage as an indicator of net land atmosphere exchange of CO2, you want to capture as much of it as possible. Typically, the dynamics of carbon at depth are, it turns over quite a bit slower when you get down below 60 centimeters. That said, if you have, an alfalfa grass in your cropping system, then you're going to need to account for carbon stocks in those deeper layers. So, and of course, the deeper you go, then distinguishing between carbonate carbon and organic carbon becomes also very important because the dynamics of those two forms are fundamentally different.
So. And I have one other question as well. So with respect, I guess, to carbon cycling, nitrogen cycling, and certainly in the context of canola, it's not a question. So in terms of priorities in terms of greenhouse gas mitigation and the canola context, what are the current priorities for canola, and perhaps looking forward 5 to 10 years?
Yeah, I see, especially being in agriculture, the N2O is it's a big problem because N2O is a potent greenhouse gas. Our canola is very productive, and we can't afford to not give the crop nitrogen. But if we put too much fertilizer on, we've got all kinds of off-target leakage effects, and N2O is one of them. And that's where there's going to be increasing scrutiny on leakage of reactive N in general from canola cropping systems, but also especially the N2O part. I would see a big priority. Everybody gets excited about the carbon because the residue inputs in a canola system are high relative to other crops, and that's a great thing. But your CO2 inputs and outputs will eventually equilibrate. But to maintain that productivity, if you've got even a kilogram or 3 of N2ON linking every time you follow a canola crop, that greenhouse gas footprint is going to come back and bite you.
Yeah. Just a quick question. One question just come in from Wayne Truman. Why are the nitrous oxide emissions higher in black soils compared to brown?
Yeah, that's mainly because of productivity is one factor. We generally have larger canola crops in the black soil, and we've got more nitrogen cycling. We've got greater crop and uptake. We've got greater residue loads decomposing nitrogen with both N mineralization and immobilization, as Lyle Cowell pointed out earlier today, happening. And that's much greater in the black soil zone where there's more moisture. In the brown soil zone, it's drier and the biological activity is less, as is plant growth. So that's why we get the difference.
Okay, thanks, Ben. I have a question also for John. And that's, I guess, a question from me actually, with respect to the yellow index and yields and the correlations there. Have you looked at the impact of canopy temperature and how that influences? Because obviously canopy temperature can impact fertility in canola. So has that been sort of looked at in terms of a co-variable when you're doing the analysis?
I have taken thermal imagery of the plots, but not for the yield estimation aspect of it, because I've mostly done this in dryland environments where there's a lot of exposed soil variation, and the variation in soil temperature causes elevations in canopy temperature. And so I can't get a clear canopy temperature reading.
Okay, thanks. And in terms of the work that you obviously presented and published, in terms of I guess leveraging that in or translating it to utility in a wider context, is that something that you also maybe in your previous roles, that's something you're involved in, but it's something that you're looking at now?
Right now I live in Ontario, and there's not a big market for canola research here.
Right.
But in other places, yeah, it's taken up some adoption and phenotyping, I think primarily, and we've had people cite us. Some European researchers where rapeseed is important. They found it to be useful for estimating when you hit maximum bloom, and things like that. But I mostly we started looking at this topic as part of a USDA-funded grant from the National Institute of Food and Agriculture for using oil seeds as a feedstock for renewable biojet fuel. And so just my main task I was given was to come up with a mid-season yield estimate for canola based on satellite imagery. And in doing that, we happened upon, I developed that index mostly to try to find out where canola fields were, because oilseed crushing facilities are interested in knowing inventory of canola production in the immediate vicinity. And so that's really how the Ellert index started, was trying to use satellite imagery to find out where the fields were and how big they were, so that the oilseed crushing facilities would know how much feedstock to expect.
Okay, thanks a lot. I think we're at the end of the Q&A session. We're almost at 3:30. So I'd like to thank Ben and John for the presentations. They were great. I think there might be some final comments from Warren Wood. Oh, I should add that additional questions can be answered in the networking session, which starts at well, I guess in a minute. But I guess I'll hand over to Warren now for some final comments.
Great, thank you, Andrew. And I guess as our presentation portion of the day comes to an end, I'd just like to make a few closing comments here. So I'd like to take this opportunity to thank all of the presenters and attendees for attending the Canola Discovery Forum of Canola Week 2020. Fertility is one of the key areas that we see driving the canola industry towards our goal of 52 bushels per acre. And your participation in events such as the Canola Discovery Forum will help move the industry there by increasing for our fertility practices, sustainability, for this increase in production. So thank you all for your participation today. I would all like to thank our sponsors for this conference. Our gold sponsors are Alberta Canola, BASF, Bayer, Cargill, Manitoba Canola Growers, and SAS Canola. Our silver level sponsors are the Canadian Canola Growers Association, the Global Institute for Food Security, and Western Grains Research Foundation. Our bronze sponsors are Cantera Seeds, DL Seeds, the National Research Council Canada, and New Seed. A special thanks goes out to AgWest Bio, and we would also like to thank the core funders of the Canola Council of Canada for their support. Before we end the day, I would like to also mention that the presentations will be available on demand for 1 week following the conference. So if you happen to have missed something or you wanted to go back and rewatch something, you will have the ability to do that. At this time, I would invite everyone to join our presenters and other attendees in the networking lounge. And again, you can enter that networking lounge by going back to the main conference lobby page and entering the network lounge from there. I would like to thank you for joining us on this virtual journey of canola fertility, and have a nice day. We'll see you in the networking lounge.